Okay, well, thanks for being here, for Abby's defense, here virtually and in person both. This has been quite a journey, and it's ended in a very good spot, which we all like to see. Abby graduated from Yale in 2002 and worked for EPA, worked on toxic cleanup and worked on Hurricane Katrina aftermath. And I think it's okay to say that probably that wasn't your niche. And I think she saw the value of a master's degree, so then came to San Diego State and to do the work that she's going to talk about today. She then joined, she left San Diego State to take a Sea Grant Fellowship in 2020 with the Energy Commission, and then joined the Department of Interior Bureau of Energy Management, Ocean Energy Management in 2021. So it's really a testament that you got this all done, being fully employed, and unfortunately it doesn't always end this well. And so I really appreciate your persistence, and also Matt's help and his lab's help in getting this shepherded through across the finish line. So the committee is happy with her thesis. There's a paper about ready to be submitted. So we're really good there. She's now working for the Bureau of Ocean Energy Management. She's working on renewable energy projects and supporting environmental studies programs. And she's going to be doing a research cruise next week. So we got this sandwiched in with Noah. That's great. I do want to say that Abby didn't walk into an ongoing research project. She really had to design the chambers, purchase equipment, get it working. She had a lot of help in that from Melissa Ward, who's a former PhD student, Alexandra Karsh, again, Matt Edwards in his lab, GCRG, but it was not a kick in the walk in the park project, right? I think it's fair to say, but the important thing is she got it done, and the paper is going to be a good paper, a really nice contribution. And so with that, I'll turn it over to you. Okay. And Trent, her third committee member, is online. Okay, yeah. Thanks.
All right, thanks for the introduction. And thank you all for coming today. So I'm going to be discussing my research, the contribution of eelgrass community metabolism to the carbon flux of San Diego Bay. I was originally interested in this research because I knew I wanted to work on climate change. I feel that it's an existential crisis for humanity. And as I was looking around, it seemed that, at least to me at the time, a lot of research on climate change in ecology was documenting the negative sequelae of climate change on various organisms and ecosystems. And this project appealed to me because it was solutions oriented, an investigation to see whether eelgrass could be a local solution for a global problem. That's what brought me here. Climate change is real. In fact, there has been a 50% increase in atmospheric carbon dioxide since 1970 from industrial activities. On the left is the Keeling curve. This is the atmospheric carbon dioxide levels at the Mauna Loa Observatory in Hawaii. And in 2013, we reached an ominous milestone where the atmospheric carbon dioxide bypassed 400 parts per million for the first time since the Pliocene. Fortunately, the ocean helps to ameliorate the rise in greenhouse gases by absorbing up to 30% of anthropogenically produced carbon dioxide. In the deep ocean, this happens through the activity of photosynthetic algae. Along the coasts, certain habitats play a key role by sequestering carbon in underlying sediments, and this is called blue carbon. Marine vegetation is functionally similar to terrestrial vegetation by removing carbon dioxide from the atmosphere. And 4 coastal habitats provide carbon services. Salt marshes, kelp forests, and seagrasses, as outlined in green, have high primary productivity due to their rapid carbon processing. This vegetation can contribute to blue carbon by net export of plant material, such as when kelp strands are dragged from the coast and buried in deep ocean waters. Seagrasses and mangroves, outlined in blue, have high carbon burial and underlying sediments due to the low breakdown of organic material. Note that seagrasses are at the intersection of both of these carbon services. So how does subaquatic vegetation take up carbon? There's 3 stages. First, carbon dioxide is dissolved from the atmosphere into the water column. Then it moves from the water column and is metabolized by vegetation during photosynthesis, releasing oxygen. And finally, organic carbon is buried in sediments and stored for millennia, as long as these beds are not disturbed, with an emphasis on the beds not being disturbed. So some keywords I'll be repeating throughout this presentation is autotrophy and heterotrophy. An autotrophic community absorbs more carbon than it releases. It is photosynthesis dominant. While a heterotrophic community releases more carbon than it absorbs. It is respiration dominant. This slide is a closer look at seagrass metabolism and the interplay of photosynthesis and respiration. During sunlight, seagrass draws down carbon dioxide and releases oxygen, as shown on the left, resulting in high net community productivity and carbon uptake. Respiration on the right is the reverse of this equation, the uptake of oxygen and the release of carbon dioxide. Respiration happens at night, throughout the day and night, but it predominates at night and during senescence or plant death. This results in carbon production and decreased net community productivity. This slide just shows the eelgrass plant, but the metabolism that we studied was at the community level. And a community consists of the dominant subaquatic vegetation, in this case, eelgrass. It also consists of the epifauna, or animals living on the surface of the seabed, like snails, infauna, or animals living on the sediments of the seafloor, like clams, plankton and algae, such as seaweed floating in the water column, and the microbiome, consisting of bacteria, viruses, and fungi. While a plant is always net autotrophic, the community in which it resides may not be because of the contribution of other species in this community. And environmental conditions can also affect metabolic status. When the entire community is autotrophic, it is photosynthesis dominant and absorbs more carbon than it releases. And when the entire community is heterotrophic, it is respiration dominant and releases more carbon than it absorbs. Now to give you a background on the plant I was working in. So eelgrass is the dominant marine vegetation within the bays and estuaries of Southern California. In San Diego Bay, it grows along the edges and in the southern border in the absence of dredging. Unfortunately, about 90% of the region's coastal wetland habitats have been lost due to development, eutrophication, and dredging. Eelgrass is a valuable ecosystem due to the ecosystem services it provides. In fact, NOAA Fisheries has designated it a habitat area of particular concern, or HAPC, meaning it provides extremely important ecological functions and is especially vulnerable to degradation. This designation marks it as a high priority for essential fish habitat conservation. So it acts as a nursery by providing habitat for spawning, breeding, feeding, and growth of many species of commercially important fish. It also plays a key role in stabilization by attenuating wave energy, allowing for sediment deposition, and both of these protect the coasts against storms. It is also a filter by providing oxygen and removing pollutants from the water column. This improves water quality. It's also important for migratory birds by acting as a stopover site and providing food source. I was interested in learning whether eelgrass in San Diego Bay can also provide blue carbon ecosystem services. Now moving on to my research. I had 3 research objectives: to determine the net metabolism and carbon flux of eelgrass meadows in South San Diego Bay, to compare the metabolism of the eelgrass meadow to unvegetated habitat, and finally to look for patterns of seasonal variation. This shows my study site, so I'm located in Southern California. The middle map is a map of the eelgrass coverage in San Diego Bay. As you can see, the majority is in the south end of the bay. And I want to draw your attention to this deep line of demarcation here that comes off the Sweet River. What is it? Sweetwater River, thank you. So as you can see, it looks like there's an artificial line of where the eelgrass stops growing. And that is because that is the range for dredging. So north of that line, San Diego Bay is dredged for navigation, and it is not dredged south of that line. And then the far right picture is a close-up of my site. So my site was at Bayside Park, which is near the Chula Vista Marina, and the eelgrass meadow I investigated is represented by this green rectangle. I used incubation chambers, which isolate specific areas of the benthos containing a known biomass. Due to the unique hydrodynamics of eelgrass beds and a limited budget, I designed and constructed collapsible chambers, which were large enough to reduce chamber effects. They had flexible walls, allowing wave energy transfer to prevent a boundary layer formation. Chamber effects and boundary layer both relate to the premature cessation of photosynthesis if all the carbon dioxide is used up in either a small volume chamber or in the microenvironment of the water column immediately surrounding the thalli or blade where photosynthesis occurs. The walls were made of PVC vinyl, which allowed for full transmission of light. And in fact, you can see how the walls practically disappear. On the right is my field setup. 3 eelgrass chambers were haphazardly placed over dense eelgrass strands, while 3 unvegetated chambers were placed over nearby unvegetated soft sediment for most months from September 2018 to August 2019. In total, I was able to do 9 months of deployments out of the calendar year. 2 of the missing months were during finals when I was not able to gather enough volunteers on scuba. And the other month it rained every week, and you can't dive in the bay after it rains due to runoff. Dissolved oxygen sensors were placed inside each chamber, and these take minute-to-minute readings of the oxygen levels inside the chamber and also provide the seawater temperature. We took water samples at dawn, dusk, and dawn the next day to calculate total inorganic carbon, or TIC, from titrations. Because the community was enclosed, any measured water chemistry changes were a result of the community metabolism within the chamber. Note that the metabolism captured included the entire community within the chamber and not just the eelgrass. This is important because, as I said before, a plant is always net autotrophic, but as eelgrass is a foundational species, the community it hosts may not be. PAR, or photosynthetically active radiation sensors, were placed outside the chambers, and PAR is the spectrum of visible light that plants can use for photosynthesis, which is 400 to 700 nanometers. We also took biomass samples from inside the chamber and took the samples back to the lab to get dry weights. This is an example of what raw dissolved oxygen data looks like from a trial deployment in August 2018. PAR data is shown in orange. And the blue line shows the oxygen sensor reading. Daylight is in yellow, and this middle box is nighttime when there's no sun. You can see that PAR follows daylight hours, and oxygen production follows PAR with some hysteresis. This formula shows how net community productivity is then calculated from oxygen readings, where V is the chamber volume, A is the benthic area covered by the chamber, and T is the incubation time. Metabolism studies examine net community productivity, or NCP, community respiration, or CR, and gross community production, or GCP. There are many ways to define NCP. One is that NCP is the net amount of carbon removed from a system via the biological pump. An analogy to your pay stub can be helpful to understand these three parameters. NCP is your take-home pay, GCP is your gross pay, and CR are the taxes taken out of your pay. So as I said, NCP was derived directly from dissolved oxygen data with its formula. Community respiration was calculated by getting the nightly hourly average and then multiplying it by 24. And you can do this because during daylight hours, both respiration and photosynthesis are going on, but at night, only respiration is going on. So you look at the total nighttime respiration, get the hourly average, and then multiply it by 24. And then finally, GCP is calculated by moving this equation around to get GCP is the difference between NCP and CR. So that was my oxygen data. Moving on to how I got my carbon data. So as I said, we took 3 water samples at sunrise on day 1, sunset on day 1, and sunrise on day 2. So this here shows some of my divers hooking up a tube to the port on the chamber. The tube is then taken to the top of the water surface and water samples are drawn with a lure lock syringe. We cleared the water several times to make sure that the water being drawn up was taken from the interior of the chamber. The bottles were then kept in these highly scientific apparatus to keep them safe. And as there were 6 chambers, I took my flotilla of floats around the various chambers and collected my samples. These water bottles were then poisoned to stop the metabolism from occurring and taken back to the lab. At the lab, they are run through a titrator. So this is a machine that calculates the different speciation of carbon. Takes a few hours, and at the end, you get this output, which gives you, it adds up all of the carbon species to give you the total inorganic carbon. Moving on to my results. So first, I'm going to discuss community metabolism, which again was derived from the DO data. So I'll be showing 3 graphs on this slide, but just to start with the NCP. So this is the net community productivity. The value for NCP is on the y-axis. The month is on the x-axis. The eelgrass chambers are shown in gray, and the non-eelgrass habitat, or NEH chambers, are shown in white. A negative value indicates heterotrophy, or net respiration, while a positive value reflects autotrophy, or net production. So as you can see, both habitats through all months of the year were heterotrophic. This is the GCP, the gross community productivity, and we found that GCP was 91% greater in the eelgrass meadow than in the NEH chambers. And here is the community respiration, and we found that the community respiration was 70% greater in the eelgrass meadow than in the NEH chambers. So overall, you can see that production and respiration were more extreme inside eelgrass meadows than in non-eelgrass habitat. And this makes sense because the eelgrass community is more dynamic than bare sediment. We also looked at seawater temperature and found that it played a key role in community metabolism. So we found that monthly differences in NCP and CR were statistically correlated with seasonal changes in seawater. In terms of NCP decreased as temperature increased, and CR increased as seawater temperature increased. I was unable to transform the GCP data to meet statistical assumption requirements, but the general trend was that GCP increases with temperature. So you can see that higher temperatures increase respiratory demand on eelgrass, and this affects NCP and CR values. And this makes sense when you think about what happens to your own body when it gets hot. It gets hot and you breathe harder, and that's what the eelgrass is doing. The respiratory demand increases as the temperature goes up. I took this data to analyze the GCP to CR ratio, and this ratio is particularly useful because it shows the balance between respiration and primary production. So the ratio is GCP over CR. When this value is less than 1, it indicates that the denominator, or respiration, is greater than the numerator, or production, and this indicates heterotrophy. When the ratio is less than 1, or is greater than 1, this indicates that production is greater than respiration, and this indicates autotrophy. So on the right are the results with eelgrass on the left and NEH on the right. And this dotted line here is where the metabolism would be perfectly in balance, where production would equal respiration. As you can see, all of the chambers throughout the year all fell to the left of the dotted line, further confirming that this is a heterotrophic ecosystem or community. Okay, so that was a lot of data. So to take a breather, show some fun field data pictures. And now I'm going to move from my oxygen data onto my carbon data. Okay, so now I'm looking at the change in total inorganic carbon over the course of each deployment. This, and again, I'll be showing 3 graphs. So this first graph up is the diurnal change in total inorganic carbon. So this is basically the change in carbon that occurs from the start to the end of the deployment. So if in terms of my bottle samples, that's the 3rd bottle sample minus the 1st bottle sample. A positive value indicates carbon released from the community to the environment. And a negative value indicates carbon is being drawn down. You can see that for the most part, the chambers produce positive values, but the trend, it was lessened and even reversed in the winter and early spring months. And that's because the seawater temperature is lower, and this is the growth season for eelgrass. So they're taking in more carbon to build up plant tissue. I broke down the total change into day and night values. This is the change in TIC during daylight hours, so from sunrise to sunset. You can see that the values are negative, again indicating that carbon drawdown happens during the day when photosynthesis is occurring. And at night, this is the change in overnight from that 2nd bottle to the 3rd bottle, sunset to sunrise. When considered over the entire year, average diurnal carbon fluxes varied significantly among sample months, but not between the 2 habitat types. And integrated over the year, diurnal TIC was larger in the eelgrass meadow than in the unvegetated habitat, suggesting the eelgrass meadow was a larger daily source of carbon dioxide. I also looked at the role of temperature in carbon dynamics. And similar to metabolism, we found that temperature played a key role. So over a complete diurnal cycle, carbon dioxide production was positively related to temperature. At night, it was also positively related to temperature. And during the day, it was negatively related to temperature. And again, this shows that increased temperatures, increased energy demands on the eelgrass plants. Another fun picture break of fieldwork before I show my last graph. So in my last graph, I compare my oxygen data to my carbon data. So I knew the timestamps of when I took the bottle samples, and I matched that up with the oxygen values on my dissolved oxygen sensors. So I was able to compare the exact time frame to look at how the change in oxygen affected the change in carbon. And we found that when examined within the same chambers and over the same time periods, changes in dissolved oxygen were significantly correlated to changes in total inorganic carbon. At night, the correlation was 67%, and during the day, it was 58%. And what this means in English is that, for example, at night, 67% of the change in carbon is related to the change in oxygen. As you've seen, getting carbon data is a lot more work intensive and with a much lower resolution than getting oxygen data. As oxygen sensors are easier and less time consuming to work with, and they provide minute-to-minute resolution, this strong correlation indicates that future carbon flux studies can rely solely on the former methodology to make inferences about carbon fluxes. Next, my discussion. So number one, we found that respiration exceeded production in both habitats throughout the year. They were heterotrophic and thus sources of carbon dioxide throughout the year. When compared, we found the eelgrass meadow was more metabolically active and thus a larger source of carbon dioxide than the unvegetated habitat. When just looking at net community production, we found it was not consistently larger or smaller in the eelgrass compared to the non-eelgrass chambers. And this is because the relationship between GCP and CR varied throughout the year. Number four, we found that community metabolism and carbon flux dynamics varied seasonally, coincident with changes in seawater temperature. And finally, we showed that changes in dissolved oxygen and total inorganic carbon were strongly coupled, indicating that carbon flux dynamics can be estimated using more cost-effective data loggers. So overall, we found that contrary to the idea that eelgrass is a blue carbon source, they were heterotrophic throughout the year. Why might this have been? So to give you a hint, all the pictures that I've shown in my presentation thus far were not from my site, they are from Google, and this is what my site looked like. So you can see it's very pea greeny and soup water. So this picture here shows an algal bloom floating in the water. This next picture here, notice this matting on the benthos, practically choking the eelgrass. This picture here is a close-up of the phalli or blade of eelgrass, and you can see how much algal epiphytic growth is on the eelgrass phalli. And an epiphyte is a plant that grows on top of another plant. So you may ask, if you have one plant growing on top of another plant, why doesn't the productivity of the algae just replace the lost productivity of the eelgrass? And we actually found that this is in line with other studies that found that dense associations of seaweeds can be respiration dominant. And then finally, in the last picture, these branching brown white structures are bryozoans. Bryozoans are a simple animal that actually calcify the eelgrass strands, and this also stops their metabolism. So in summary, we found that the low productivity was a combination of increased respiration and decreased photosynthesis.
So to walk you through this, first of all, South San Diego Bay has very long residence times. In fact, previous studies found that it takes 60 days for water to cycle in from the mouth down to South San Diego Bay. This is in comparison to the mouth of the bay, where water cycles with every tide or every 6 hours. So the very long residence times means there's little flushing or water turnover. South San Diego Bay is very shallow, allowing for a lot of evaporation. And we're in a Mediterranean climate, there's minimal freshwater input. So the combination of little flushing, shallow, with minimal freshwater input creates warm, hypersaline waters, and this stresses the eelgrass out. This causes increased respiration. On the right side of the graph, my site was at Bayside Park in Chula Vista, and which specifically in general San Diego is an urban environment. It's a densely populated coast. When you have a lot of people, it causes sedimentation and eutrophication. And eutrophication is increased pollution from runoff. The eutrophication leads to algal blooms and epiphytic loading. The combination of eutrophication and sedimentation also causes increased turbidity, i.e. the water is not clear. So all of this leads to low light levels in the water where the eelgrass lives. And as the amount of photosynthate that a plant produces is directly correlated to how much light it receives, if the plant receives less light, you're going to have decreased photosynthesis output. So in conclusion, this environment causes increased respiration with decreased photosynthesis, with the result being lower productivity. So what were my conclusions, my takeaway points? Unfortunately, eelgrass meadows are too stressed to be net carbon sinks in Southern California, although they are able to be where the water is colder, such as our colleagues in Davis that found their carbon sinks in Bodega Bay. However, when you look at a scientific study, you need to understand exactly what is being measured. So carbon flux studies may not capture the entire story. So as a refresher from the beginning, carbon studies can focus on 3 different fluxes. The first from the atmosphere to the water column, the second from the water column into the plant, and the third from the plant into underlying sediments. And my study focused solely on metabolism. So despite my data showing the eelgrass meadows were sources and not sinks, that was only examining the metabolic carbon in the system. They can still sequester carbon in underlying sediments. And the fact that these systems even exist mean that they are storing carbon in plant tissue. So removing the meadow would release all that carbon contained to the atmosphere. So while eelgrass meadows in San Diego Bay may not be blue carbon reservoirs, they should still be preserved for their many ecosystem services. So eelgrass habitat is protected by laws like the Clean Water Act, the Magnuson-Stevens Act, the California Coastal Act, for all the reasons shown in this graphic. And what these laws mean is that eelgrass habitat loss must be mitigated by either restoring disturbed land or creating new habitat of equivalent acreage lost. So if eelgrass habitat is disturbed by dredging, it needs to be replaced. And I saw this happen in practice during a practice deployment in Mission Bay. I saw dredging occurring where these large ships come around and they scoop up the top layer of the eelgrass. They dredge out the underlying sediment so that the channel navigation is maintained. And then they plop that eelgrass habitat back down. This does preserve a lot of the ecosystem services of the eelgrass. It still is able to be a nursery, feed birds, improve water quality. However, Eelgrass cannot be considered a blue carbon source if the underlying sediment, the reservoir for carbon sequestration, is constantly disturbed by dredging. But we need to be realistic because San Diego Bay is a working harbor and it will continue to be dredged. So the answer, I would say we should recognize that this ecosystem is a whole system and that everything is connected. And we have made some progress. For example, in the 1950s, over 50 million gallons of sewage and industrial waste were dumped directly into the bay. And we're not doing that anymore, so progress. And there is a lot of less eutrophication in the bay than it used to be. So I think it's important to continue to educate and value the ecosystem for the services that it does provide. With that being said, I would like to thank many people for their help in my project. This is definitely a marathon and not a sprint. First and foremost, I have to thank my volunteers. I had between 30 and 40 people help me out on this project, ranging from scuba divers, people on snorkel, people helping me process the eelgrass back at the lab. I had students help construct chambers. I spent 6 months experimenting with different types of glue to find one that would not work in salt water, not degrade the PVC vinyl, and create an enclosed environment. And I had students helping me with that every way. So the project wouldn't have happened without my volunteers. I'd also like to thank the labs that I was part of, the beer pigs, the benthic ecology, experimental research, phycology in general, and global change research group labs, whose students and PIs helped me a lot throughout this process. CMIL, the Coastal Marine Institute and Laboratory, is an invaluable resource. So many thanks go to the people there. SDSU Biology, my committee, my colleagues at BOEM who continue to encourage me to finish this out. And finally, my family and friends. And this is a little painting I did actually at Restore America's Estuaries. They had an art session. So this is a representation of my work that shows the photosynthesis equation. That being said, I'd like to take any questions.
Thanks, Abby. This is a thesis defense. So I'd like to first ask, Trent is on, good. Trent, can you hear me and can you speak?
Yeah, can you hear me?
Great, yes, perfect. Matt, do you, okay, let me ask, Trent, I'll ask Matt if, for his comments and questions, and then I'll ask you, okay?
Yep.
First, Abby, I think you did a really good job of taking a very complicated story and cutting through all the data and making it into something simple. However, I'm going to reverse that and make this a little more complicated for you right now. And you know this question's coming. I kind of let you know it's going to come, but I'm also going to throw you a curveball for follow-up. So if you're talking to the Port of San Diego, who have a big endeavor right now, and some of them may be on this, for restoring eelgrass for blue carbon, not for ecosystem services, but for the blue carbon. You tell them it's respiration driven, which tells me maybe they shouldn't restore these beds from a carbon standpoint. What would you tell them?
Okay, well, I did let the port know. I don't know if they got my message or not because they're kind of hard to get a hold of. But basically what I would say is that in the navigable part of the bay, if you are dredging the bay, eelgrass is not going to be able to store, is not going to be able to sequester carbon in underlying sediments. So if you want to look at the sequestering capability of eelgrass, you really need to focus on the South Bay and emphasize that those aren't dredged now and they shouldn't be disturbed in the future. My colleague Melissa Ward, she looked at all three fluxes, and she did look at the carbon burial capability, and it takes something like, I don't remember the results, but it takes like 500 years to really create a good carbon load in the soil. So if you want to use eelgrass as for blue carbon in San Diego Bay, you need to not disturb it.
Good, and then the curveball.
Okay.
So your story is about inorganic carbon. CO2 bicarbonate, essentially right? We haven't talked about organic, you've mentioned it a little bit, but I'm wondering if you think that the taking up and the turning inorganic carbon into organic carbon, that exceeds the off gassing. That is, you have more carbon going into organic forms as organic through primary and secondary production, right? If that could be, so you might be picking up this off gas and this net respiration, but we're not measuring how much carbon, how many moles of carbon are being accrued into organic forms. If that exceeds what you measure in inorganic, might it be that these things actually might actually be net autotrophic? We just don't pick that up with community metabolism. Community metabolism is how we do this stuff very often. But what do you think about the organic side?
Well, so what I would say is that the plants themselves are definitely autotrophic. I would also say that what I've learned at BOEM is to not speak outside your expertise. And I am not a subject matter expert on organic carbon. So I would have to say I don't have a response to your question. I don't know. It's hard for me to estimate. I mean, I don't know what the relationship between organic and inorganic carbon is, so it would be complete conjecture.
Yes. Do you think do you think a lot of carbon is going into organic forms?
I would say probably, yes. But again, I could be wrong. Don't quote me on that.
A true government employee. Following up on what Matt said, I would like to think of this maybe from a different perspective. And these systems cannot be losing carbon forever, right? It's impossible. So what is let me first ask you what your explanation is. How can they be net sources of carbon?
Okay. Well, you know, we did show that their productivity is strongly correlated to seawater temperature. And unfortunately, seawater temperature is on the rise. One of my deployments, I think like in August or something like that, at that time, it was the hottest temperature ever. And normally when you dive, you wear 70 mil wetsuits, and I went in with no wetsuit whatsoever. And my volunteer divers were like, this is like diving in a bathtub. So when you have increasing seawater temperatures, you are going to be losing carbon into the atmosphere, because disturbing an ecosystem negatively affects its ability to function and provide those ecosystem services. So I don't know if that means that eelgrass may eventually disappear. I know that right now, eelgrass habitat does grow into the northern part of Mexico a little bit, but we are sort of at the southernmost range of where it likes to exist. And it's like a different species in Bodega Bay. It's so much healthier. The blades, the thalli are a lot thicker. They're a lot taller. So maybe as seawater temperature rises, that like with a lot of other species, the ranges will be shifting north. I don't know, but
yeah. It could be that it's a transitory effect, as you say. It's gotten warmer and there's more respiration, and that's why it's negative. It could also be that we know there's an exogenous source of carbon, right? From runoff. And so, and that's something you haven't looked at. It's not factored in, but it's entirely possible. In fact, I think it's probable that what we're seeing is the effect of a big input of carbon into that region, and some of that's being respired. You sort of referred to the plants, but you're also measuring the soil respiration and the soil microbes. So my guess is that the system, you're seeing what you're seeing because of the exterior source of carbon to the system.
I'm just checking because I know Trent has to leave
At what time?
at 11, I think.
Yeah.
This is good.
So I would be really careful about your message, and I would think about rephrasing it, and I would think about the net effect, because your loss to the atmosphere is smaller in the seagrass dominated than in the non-seagrass dominated systems, right?
Well, we found there was the carbon fluxes were larger in the seagrass beds because it was a more dynamic community.
I think the net fluxes are smaller, if I remember your data correctly. We can talk about this more, but I would be very careful about the message, because this is a system that's heavily polluted. There's a lot of exogenous carbon that's getting undoubtedly burned off by the soil and in the water column. And if the message that's received at San Diego Port Authority and others is that there's no blue carbon benefit, that we may have, if it's true, it's important to get that out. But if it is an effect of the metabolism of exogenous carbon, then we need to rephrase it. One thing I would ask you, you said it's a working harbor and the dredging's necessary. Is all the dredging necessary? For example, off of Silver Strand, is that really necessary dredging?
You know, I did try to find out exactly the dredge schedule and where it happens and how often it happens, and I was never able to get that information. So I'm assuming they just draw a line and said, we're gonna dredge to the north of here. So it could be that, you know, deeper analysis, spatial modeling, which we're familiar with at BOEM, could be done to identify other areas of the harbor which don't need to be dredged.
Mm-hmm. Yeah, great, thanks. Trent, do you have comments and questions?
Yeah, I just would say terrific job with a lot of field work. Your presentation's very clear. I would just echo the need to recognize that the eelgrass can't keep emitting carbon forever without some input of carbon coming from somewhere, either from the river system, or is it possible that there's production of algae in the water column above the that would be above the height of your chambers that has created that has resulted in organic matter sedimentation, and you're seeing the respiration of that accumulated algal biomass? That's one question. And then another sort of note on interpretation, you said maybe this was a sort of a transient effect due to high temperatures. It's almost like a coral bleaching event where these systems are being exposed to stressors they've never seen. So it could be a very temporary phenomenon. And then just sort of as a footnote there, in your conclusion, you said, well, Northern California eelgrass may sequester carbon, maybe Southern California ones don't. But I would also add some qualifiers in highly eutrophic, urbanized watersheds, just to make sure that the message is clear there. So those are my main comments.
Okay, I'm gonna address the second one first, because that's what is on the top of my mind. So I do think eutrophication plays a large part, but the temperature of the water is even more important, and that's why I made that sort of blanket statement about Southern California. And the reason for this is that in March of my deployment year, we did do a paired deployment. So we did one at Chula Vista. We also did one at the reserve in Mission Bay, which I can't remember the name off the top of my head. But this picture here is actually from the Mission Bay. And so you can see that the water does look a lot better and the eelgrass does look a lot healthier. But despite that, the results were still heterotrophic, even in Mission Bay. So I think the temperature of the water is really important. And sorry, I forget what your first question was.
I was wondering if where the, sort of similar to Walt's question, where is the carbon coming from? It has to be coming from somewhere.
Yeah, about
And
yeah,
maybe it's coming from dead algae settling on the.
I would definitely agree with you on that. Especially in the hot summer months, the water, like the turbidity was crazy, and a large part of that was just how much algae there was floating in the water. Like there would just be beach ball sized blooms of algae in the water. And even if I didn't need to wear a wetsuit because the temperature, I would wear a skin because I was getting stung all the time from whatever it was in the water. There was a lot of stuff floating in the water when the temperature went up. So I do think that algal respiration definitely contributes.
So in the end, if you integrated the whole water column, the whole system could be a carbon sink. But it's just that the benthos and within a meter of right to the top of the seagrass canopy is a source, because you're respiring the benthic carbon that accumulated from the algal biomass settling on the bottom.
Yeah, I do
possible.
think the chambers were pretty representative of the water column because they were very big. They're probably bigger than they needed to be. I had to be careful with picking what part of the month I would go out, because initially my chambers actually emerged because the chamber was taller than the height of the water. So afterwards I always made sure to go out during neap tides, so there wouldn't be as strong a difference. So at times, like the chamber was the total height of the water.
Great. Yeah, thanks. I have some additional comments in the thesis I'll send you. Great job.
Thank you.
So, sorry, Trent, you said you do have additional comments or none?
Just a few. Yeah.
Do you want to make them now?
No, they're just on the thesis document
Oh,
itself.
okay. All right. Kevin? Yeah.
Yeah, I'd love to ask. Nice job. I'm really glad you did the study.
Thank you.
Yeah, it's great, but I do take issue with a couple of things you said. On slide 29, or maybe I thought it was a different one, actually, a summary one. Oh, no, it is right. What you have up there is perfect.
Oh.
Yeah, eelgrass meadows are too stressed to be then carbon sinks to Southern California. You studied one site, notwithstanding maybe Mission Bay, pilots, whatever it happened to be. And knowing San Diego Bay, like they do, can stress how different Chula Vista is from most of the rest of the bay. The amplified load, the rise zones, particularly you pointed out, I've seen maybe one other site in Mission Bay that's like that. Otherwise, it's really, really a fundamentally different type of arrest than the other areas. And I think you're minimizing the possible importance of the other areas in the bay. The whole eelgrass. I know you look at a map of eelgrass distribution from Merkel, and it looks like the little tiny lines of green along the shorelines are insignificant, but those are actually pretty well established and large eelgrass beds. Much different type of environment than Chula Vista in the south part of San Diego Bay. I think they contribute collectively quite a lot to carbon sink basically into the bag. So that the light levels are higher, the amplified production is higher, the amphipod and Aquinas diversity is much higher, the trophic transfer rates are probably much higher, and they're tidally influenced. So I think there's a lot of export of carbon probably out of the bay from those places. So I wouldn't minimize those areas just because it looks small on that.
Yeah,
I wouldn't extrapolate to Southern California. I think that's dangerous.
Okay, yeah, no, I see the point, and I do think ecology is sometimes the story of trying to take small studies and extrapolating and make broad ecological statements. So I agree, I should be careful about that. And I did try, or initially, my goal was to kind of do a paired study in either Shelter Island or Coronado Bridge. And I did a couple trial deployments there. And in order to make that successful, you have to use much smaller chambers. What I found, because it is navigable chambers, every time a boat went by, the chamber would completely lift up out of the ground. So even though I had, like, we were pounding the chambers into the sediment, the legs were about this long, they had skirts with chains on the skirt, like anytime a boat went by, the chamber would just lift up. So I had to do it in Chula Vista because it's, the waters are calmer there.
I understand. I think back to the experience and experiments. So, do one more thing about Northern California, Southern California. You said something about eelgrass being healthier in Northern California. That I don't see how you can make a statement like that. So eelgrass is wider, morphologically speaking, wider and usually taller in places like San Francisco Bay, other places in California, but the densities are order of magnitude lower in those areas. So the biomass is not necessarily fundamentally different. We go to places around San Diego and see densities of almost 1,000 shoots per square meter that actually are pretty long, well, pretty tall relative to the water. So I don't know how those things equate directly to health. Health is a very vague term I wouldn't use very often, personally speaking, because what does it mean exactly?
I did do biomass estimates using the same methodology from, I think it was from the Kroger lab. So I got the worksheets from Melissa and she showed me how to, run it like, and we use the exact same calculation. So I do have that data. So that data could be compared in terms of the plant material content to the Bodega Bay studies. It's just something I didn't end up focusing on.
And one last thing so you talked a lot about temperature, but what correlates with temperature in the bay seasonally is going to be light levels, right? So just not to minimize the possible relevance of light levels. The bay substantially clears up in the winter months. And you've seen this trend from some fall to winter to summer and things. So light levels change in a lot of temperatures.
Yeah, and PAR is incredibly important. Unfortunately, PAR is a lot harder to measure in that the sensors are a lot more, they're harder to work with and they flood. And so I tried to get PAR data, as much PAR data as I got temperature data. And I wasn't able to have enough months in order to do statistical comparison because of equipment failure. But it definitely, doing the study again, yes, more of an emphasis on getting more PAR measurements.
But you did get part measurements.
I got some PAR measurements, yeah.
Donatella, thanks, Kevin.
I was curious about the what, did you show any PAR relationship, any relationship with the PAR?
So I have that in the paper as a supplementary table. I didn't have enough PAR data in order to do statistical analysis.
But you saw the response to the PAR with the few data you had?
Yeah.
Oh.
Yeah.
Thanks for all the input. That was great. Are there any other questions or comments from the audience? We're pretty much at the end of the hour, but anything urgent, burning?
Chat.
Oh, there is some.
Do you want to check the chat?
Oh, no.
Just a congratulations.
Yeah.
Are there any questions from anyone on online? Feel free to come off mute if so.
Okay, if not, thank you very much for attending and for the feedback. Appreciate it. Trent, do you have to go or can you hang on for a few minutes? Or are you gone?
Checking.
Okay,
And everyone, we're gonna meet in PS 157 right after this and have a little thing. So everyone's invited.
okay, great.
Sorry.
Thanks. Thanks, Matt. Okay, thanks again. Thanks.
Okay, thank you.
Matt, Donna, Kevin, can you stay behind and good job.
Okay, thank you. All right, I will turn this off. Thank you everyone for attending.
That's all good. That's all good.
Thanks, Kevin.
It's Friday.
I just
Yeah,
need
could you
to.
just give us a.
Yeah, I'm sorry. I'm just ending the